Animal venoms have evolved to enable the capture and digestion of prey. Venom is a fluid made by an animal and injected into another animal using a fang, barb, spine, or stinger. Venoms are complex and can contain up to hundreds of toxins that can be delivered in a single dose or in multiple doses. Toxins from venomous animals are selective in their functions. For example, neurotoxins and myotoxins both aim to disable and kill the selected prey, whilst other toxins induce severe pain as a defense mechanism without resulting in the target's death. Neurotoxins target the transmission of nerve impulses and destroy nerve tissue. An example is tetrodotoxin or TTX, found in the pufferfish. Myotoxins are basic peptides found in snake venoms. They attack and break down muscle tissue, causing instant paralysis to prevent the snake's prey from escaping. Eventually, the muscles of the diaphragm are paralyzed, leading to the death of the prey. The venom in the ankle spurs of the Australian platypus is used to disable its prey. The toxin is comprised of defensin-like proteins, nerve growth factor and natriuretic peptides which induce the excretion of sodium from the kidneys. 3 of the defensin-like proteins are unique to the platypus, and although they aren't lethal to humans, they cause excruciating pain. This hyperalgesia is associated with mast cell degeneration, may last for several months and fails to respond to strong painkillers like morphine. Although venoms are usually thought of as being harmful or even lethal to humans and animals, there are several examples of drugs that have been developed from venoms, which are still in use today. For example, captopril, exenatide and ziconotide. Captopril is the first of the angiotensin-converting enzyme or ACE inhibitors. The effect of the drug on a person's blood pressure mechanism mimics those of the venom of the poisonous Brazilian pit viper. In the 1970s, researchers observed that plantation workers who had been bitten by the Brazilian pit viper collapsed shortly afterwards, as a result of rapidly dropping blood pressure. In 1975, the researchers separated the blood pressure-lowering component of the venom, modified it, and produced a synthetic version. Captopril, as it was named, achieved FDA approval in 1981. However, soon afterwards, several papers and letters reported serious hypotensive effects of the high doses of the drug that were initially recommended. By the mid-1980s, a longer-acting ACE inhibitor called enalapril was released. Initially, enalapril appeared to have fewer side effects and an easier dosing schedule than captopril. However, reports started appearing about hypotensive events similar to those experienced with captopril. This resulted in recommendations that the initiation of ACE inhibitor drug therapy should be carried out under strict medical supervision. Exenatide, a drug synthesized from the saliva of the Gila monster, has become an additional treatment for type 2 diabetes together with medication such as metformin. Gila monsters inhabit the southwestern deserts of New Mexico and Arizona in the United States, as well as the state of Sonora in northern Mexico. Although shy in spending up to 95% of their life underground, when Gila monsters venture above ground, they can be dangerous. The venom in the bite of a Gila monster can cause pain and weakness but is rarely fatal to adult humans. Exenatide is the synthetic form of a glucagon-like peptide found in the venom of the Gila monster. The peptide was discovered during research into the effects of snake and lizard venoms, which were found to cause enlargement of the pancreas, the area where insulin is synthesized. Of great interest was the fact that the Gila monster was able to slow down its metabolism and maintain constant blood sugar levels, even after long periods of not eating and without negative effects to its health. More recently, scientists have discovered that the glucagon-like peptide has neuroprotective effects in animal models of various neurodegenerative diseases. This has led to the possibility of the testing of the drug as a treatment for Alzheimer's and Parkinson's diseases. Ziconotide is a non-narcotic analgesic used for severe or chronic pain, for which all other pain relief methods have proved unsuccessful. The drug is administered directly into the spinal fluid through the intrathecal route, and is reported to be 1,000 times more powerful than morphine. However, unlike morphine, it is non-addictive. Ziconotide is derived from a pain-relieving toxin of one type of cone snail, Conus magus. When hunting for food, the cone snail shoots out doses of venom into its prey using its harpoon-shaped tooth. The first dose of the venom causes convulsions, followed by the second dose which paralyzes the cone snail's prey, allowing it to be consumed. In the 1990s, researchers at the University of Utah isolated and purified a particular Conus magus toxin that was found to block certain pain signals in animal models. The toxin was synthesized to create the drug called ziconotide, which was approved by the FDA in 2004. The pharmacological activities of venom components have been investigated as a source of potential therapeutic agents against certain cancers. The characterization of some snake venom compounds has led to studies investigating their ability to interfere with stages of tumor development, for example cell invasion and cancer metastasis. Several animal toxins display profound anti-cancer effects, in particular toxins purified from snake, bee and scorpion venoms. It is suggested that certain toxins have a similar effect on cancer cells as chemotherapy. The blunt-nosed viper, found in North Africa, the Middle East and parts of Kashmir, contains a snake venom disintegrin, a type of protein that inhibits platelet aggregation. The snake venom disintegrin has shown promise in inhibiting colon cancer cell viability. Despite this, snake venom for cancer therapy is still at an early stage. It is hoped that the combination of snake venoms with other technologies such as nanoparticles will develop into successful anti-cancer treatments in the future. Despite optimism about venom-based drugs, peptides and proteins often have crucial limitations in biomedical applications. One issue is that peptides and proteins are broken down by enzymes in the human stomach and therefore drugs based on them cannot be taken orally. This makes administration of venom-based drugs more costly than other medication. For example, ziconotide must be administered intrathecally, which is an invasive procedure. In addition, it mainly works in the central nervous system in contrast with morphine, which enters the bloodstream more easily as a simple injection. Another problem is that venom-based drugs often lose their effectiveness in the transition from animal models to human patients. For example, a drug which is used successfully on rodents may not show the same promise when used on humans. This is because venom components often lose their potency and accuracy when used as medicine in the human body.